Welcome back to another Lessons Learned video. Today, we're going to be talking about cellular respiration and going through all the steps. So let's begin. Now, the first step is glycolysis, and the two important things to remember is that glycolysis occurs in the cytosol, and the entire process is anaerobic, meaning oxygen is not present. Now, glycolysis starts with the 6-carbon sugar, glucose, and glucose will be catalyzed by 10 different enzymes. So there are 10 different steps with 10 different enzymes to produce the two products. Now, through these process, ATP is made, and there's a total of 4 ATPs being made, as well as 2 NADH, which is electron carrier molecules that will be used at the last step. Now, during this process, 2 ATPs are also used, right? And after the end of this process, the two products is pyruvate, right? And each pyruvate has 3 carbons, and these are the two products. Now, as I said, there are 10 steps to glycolysis. This is just a general overview, and the ATP made is through substrate level phosphorylation. Now, the net gain of ATP in glycolysis is a +2 ATP. Now, when the pyruvates are made, they move on to the second step, and that is the formation of acetyl-CoA. Now, 2 things to remember about the formation of acetyl-CoA is that it occurs in the mitochondrial matrix. And also, this process is aerobic, meaning oxygen is present. Now, as I said, the 2 pyruvates, which are each 3 carbons, they enter the mitochondrial matrix and react with a coenzyme A. And this reaction leads to CO2 being lost. And then further down, an NADH is made. Now, each pyruvate goes through these steps, and the production is 2 acetyl-CoAs. Now, once the acetyl-CoAs are made, they move on to the next step, which is the third, and it's called the Krebs cycle. Now, it's important to notice that acetyl-CoA has 2 carbons, right? And they react with a 4-carbon sugar, or 4-carbon molecule, called oxaloacetate. So 4, and this produces a 6-carbon molecule called citric acid. 1, 2, 3, 4, 5, 6. And this process is also called the citric acid cycle. Now, during this cycle, citric acid is catalyzed by a few different enzymes, and each time it's catalyzed, a carbon is lost. So at the end, only a 2-carbon molecule is made, so 2 carbons in, 2 carbons out. That's a quick, easy reminder. Now, during these processes, 3 NADHs are made, 2 CO2s are lost, 1 ATP is made, 1 FADH2 is made, and that's another electron carrier molecule. And this process happens a total of 2 times. Now, this is because 2 acetyl-CoAs were made, so that means each acetyl-CoA goes through this cycle. Now, the next and final step is oxidative phosphorylation. Now, this is split into 2 different stages. The first stage is the electron transport chain. And in this chain, the 10 NADHs that are made and the 2 FADH2s that are made, so the electron carrier molecules, they will end up, their hydrogens, which carry energy, will split into protons and electrons. Now, the high energy electrons from these electron carrier molecules are passed along a series of protein carrier molecules, and it will go through a chain. Now, each carrier molecule hands down the electron to the next molecule in the chain. So I'll draw here. The red circles are each the proton carrier molecules. So the electron moves through the chain until it reaches the final electron acceptor, which is 1/2 of an oxygen. Now, as I said before, electrons are made, but also protons are made. So protons will also go through this proton pump and go into the intermembrane of the matrix. So protons are made, and it goes all the way up, and this creates a gradient, right? And now back to the final electron acceptor, the 1/2 O2. Now that will combine with 2 protons, so 2H plus, and this creates H2O, which is a waste product in cellular respiration, and this is where most of it is made. Now back to the proton gradient. Obviously, it creates a gradient, so because protons are being pumped into the intermembrane, and this causes a lot of potential energy to be stored, and also creates acidic pH in the intermembrane space. Now this moves on to the second part of oxidative phosphorylation, and that is chemiosmosis. Now the proton ion built up in the intermembrane space diffuses back through facilitated diffusion into the mitochondrial matrix through an enzyme called ATP synthase. Now, as I said, ATP synthase will spin once the protons go through, and this causes ADP, which already has 2 negatively charged phosphate groups, to bond with another negatively charged phosphate, which thus creates ATP. Now, the protons will pump back in, and this will cause the end right here of ATP synthase to again spin like a wheel, and thus creating ATP. Now, a quick reminder, it takes roughly 3 protons to make 1 ATP, and at the end of this, roughly 34 ATPs will be made. Now, that's the last step, and this is a quick chart that kind of summarizes everything right here. Now, glycolysis creates 2 ATPs because 4 is made, but 2 are used, so that's a net gain of 2. The formation of acetyl-CoA loses 2 because it takes 2 ATP to move the pyruvates into the mitochondrial matrix. The Krebs cycle creates 2, and oxidative phosphorylation creates 2, and that means 36 ATP is made. Now, some textbooks will say 38 because they do not account for the formation of acetyl-CoA. Now, for the NADH, 2 is made through glycolysis, 2 is made through the formation of acetyl-CoA, and 6 is made through the Krebs cycle. None is made in oxidative phosphorylation, so a total of 10 NADHs are made. And for FADH2, it only occurs in the Krebs cycle, and it happens 2 times, so that means 2 FADH2s are made. Now, each NADH is used to create 3 ATP molecules, while each FADH2 is used to create only 2 ATP molecules. So that is the summary of cellular respiration in under 10 minutes. If you guys have any questions, please leave it in the comments, and I'll answer them. If you have any video ideas or questions in terms of biology or any other subject, please let me know. Make sure to like, comment, and subscribe.